Welcome back to Chapter 19, Part 3. Now we're going to learn all about the evidence for evolution. So as we have been going along over the last 170 years since Darwin first came up with his ideas, we've been making lots and lots of new discoveries. We don't just, think about this 170-year-old idea and say, that's good enough, right? We're continually looking for evidence for or against Darwin's ideas. And it turns out that there are 4 basic categories of data that we've been able to document that shows the pattern of evolution and how it occurs. And those 4 categories are direct observations. Yes, we can directly observe evolution. Remember, the definition of evolution is change over time. We can use something called homology, and I'll tell you all about what that means in a little bit. We also use the fossil record, and we've known about the fossil record since before Darwin, but we've had a lot of chances to fill in a lot of the gaps since Darwin. And finally, we have this concept of biogeography. So let's start in by looking at direct observations of evolutionary change. And again, yes, we biologists have been able to actually observe evolution in thousands and thousands of scientific studies. I'm just going to be able to give you a couple of them. One of the most interesting ones that your book brings up is the case of the soapberry bug. Soapberry bugs use their beak. It's not really a beak. It's like a piercing, sucking mouth part. It actually sucks on the inside of seeds and fruits. These bugs are very effective when their beak length is similar to the depth of the seeds to get to the kind of juicy bits on the inside. So this is a picture here of the soapberry bug, and it's got its beak piercing into this balloon vine fruit. So in southern Florida, soapberry bugs feed on large fruit of balloon vines, and they have very long beaks. Right? In central Florida, these bugs actually feed on the golden rain tree fruit, and golden rain tree fruit are small, and so it turns out that the piercing, sucking mouth part, the beak, as we say, of the soapberry bugs are kind of smaller when they're eating the golden rain tree fruit, and they're larger when they are feeding on the balloon vine. Now, the important thing to realize is that we have been collecting soapberry bugs for a very long time, and so you have these insects in museum specimens, and so in the museum specimens, the average soapberry bug beak was right about 8.5 millimeters. Didn't matter if it was in Central Florida or Northern Florida, that was the average. But over time, and this is how we've directly observed it, got these historical samples, and then we've got these movement towards smaller beak length. Now, this is really important to know that the golden rain tree is an introduced species in Central Florida. And so we actually know approximately when that tree was first introduced to Central Florida. So we have a time frame here also of when that evolution started. So the correlation between fruit size and beak size has been observed in other locations as well, and other places besides in Florida. And every single time, in all cases, the beak size has evolved in the populations that feed on introduced plants with fruits that are smaller or larger than the native fruits. It just keeps happening over and over and over again. We keep making these observations. So it's not like it just happened the one time. We've seen this happen multiple times. In Florida, this evolution happened in less than 35 years. And so what we know is that evolution can happen fairly rapidly in wild populations if the selection for or against a particular trait is strong enough. The next one that we're going to talk about that's an actual direct observation of evolution is the evolution of Staphylococcus aureus and drug resistance. When we say drug resistance here, we're talking about resistance to antibiotics. One of the main places where you can find Staphylococcus aureus is on your skin or in your nasal passages. And one of the common ways to treat staph infections is by using an antibiotic called methicillin. Now, you've probably heard of penicillin. Methicillin is related to penicillin, but it used to be back in the day. It used to be very effective at killing staph infections. And one of the things that the staph does is it actually causes, there's a particular strain of it that causes flesh-eating disease. And of course, I mean, that sounds gross, and it is. Literally, your flesh of your skin and your nasal passages is slowly eaten away by these bacteria. And it used to be that if you were given methicillin, it would kill the staph. But these days, we know that that doesn't happen anymore. So we're going to talk about MRSA, and that's how this word is pronounced, MRSA. That's methicillin-resistant Staphylococcus aureus. So let's get a couple slides here on how this worked. We did and have in the past, since the 1940s, basically, where it became very, very common, started using things as penicillin and methicillin to treat bacterial infections. Very quickly after staph was first start to be tried to be killed by penicillin, within 2 years after it was first widely used, we started to see that staph became resistant to penicillin. In other words, it was harder and harder to kill the staph with the same dosage of penicillin. They had to keep upping the dosage of penicillin. And so they started using methicillin instead. And of course, within 2 years of its use, the staph also became resistant to the methicillin. Methicillin is interesting because it inhibits a protein that most bacteria use to make their cell walls. And of course, if you inhibit this protein, we've talked about this a lot, it's an enzyme used to synthesize the cell wall. If you inhibit this enzyme, then you'd think that the protein, the bacteria would die. However, MRSA bacteria use a different protein for cell wall synthesis, a different enzyme. So they have been able to survive and reproduce at higher rates when they've been exposed to methicillin. So the methicillin resistant ones had this alternative pathway to make cell walls. And when we started using methicillin all over the place, the ones that were exposed to the methicillin just like, hey, guess I'm going to make my cell wall this way. They're not thinking this, of course. They're not consciously thinking this through. It's really just the ones that survived the environment that is this poison of methicillin are the ones that are now resistant to methicillin. And MRSA strains are actually now resistant to many antibiotics. This is kind of scary. I mean, let's think about it. Nobody wants to get an infection by a bacteria that you can't kill with an antibiotic. And yet, here's MRSA, the methicillin-resistant Staphylococcus aureus. They're resistant to many, many antibiotics. And we see this over and over and over again. We see that like the bacteria that causes tuberculosis is becoming more and more resistant, so that we're no longer able, in some populations, we're no longer able to kill tuberculosis. It's coming back and it's killing humans at rates similar to what it used to be back in the 1800s. Again, remember that natural selection does not create new traits, but selects for traits that are already present in the population. So in the case of the MRSA, there were a couple of different enzymes that they could use. One was the most common enzyme. This rare one was being used, and it became more and more and more populous in the population because of descent, right? Through passing down inheritable traits. Evolution by natural selection can occur rapidly in species with short generation times, like bacteria. Bacteria can go through population doubling in about 20 minutes under ideal conditions. So that means that evolution can happen pretty quickly. It also depends on whatever the current environment is. So just kind of remember that that if there is no methicillin, right, being used, then you're not going to get methicillin resistance. So one of the things that we know is that if we stop using particular antibiotics and then start using them again in maybe 10 years, that we maybe have a little window of efficacy against these bacteria that are evolving so fast. So that's just two examples of how we've been able to see evolution happening in real time. The next concept that we're going to talk about is homology. And homology is the idea that similar traits are passed on because your ancestor had them. So remember that evolution is a process of descent with modification, right? So what this means is that related species had a common ancestor. And that trait, whatever that trait is, in this case, the bones of the limbs of the organism that first evolved those traits, they are changed over time. Maybe they take on a different function, but they still have the same general structure. So we see this in this kind of homology. Again, this is similarity because you had an ancestor that had that and just things changed a little bit over time. We can see that in the legs of humans and cats and whales and bats. Basically, all vertebrates have the same pattern of bones in their arms. The example on the same pattern is that there's this one bone that connects to your body, and it's in your upper limb. In this case, where your biceps and triceps are, your upper arm. You also see the same one bone in your thigh region. So this bone in your arm is called the humerus, and if you look at the leg of a cat or the flipper of a whale or the wing of a bat, there's just this one bone that connects to the body at the shoulder region. Next, there are two bones that connect, this radius and ulna, and you see that same situation in cats. You see these two bones connected to that one bone in whales, and the same thing in bats. And then you have like a whole bunch of little bones here called the carpals, and they branch off into fingers called the metacarpals and phalanges. And you see the same thing in cats. Even though cats don't have long fingers like we do, they still have carpals, metacarpals, and phalanges. Even though whales don't have fingers at all, they still have carpals, metacarpals, and phalanges. And bats have very, very long metacarpals and phalanges and pretty small carpals, but they're still there. So you might ask, why do things that are so different, like humans and cats, whales, really whales, and bats, why do they have the same arrangement of bones, even though they have very, very different functions? And again, we have to go back to this idea of descent with modification, because homologous structures exist because the ancestor had some version of that trait. And as time went by, that trait was modified and changed, and became the current use that it has now. Again, it's not through use and disuse. It's because the organism had this ancestor, and the modifications that led to modern humans were the ones that allowed modern humans to and their ancestors to exist and to thrive. So the same thing for whales. They start with this basic platform of the bone structure, and they moved into water. And so the ones that were able to evolve these modifications are the ones that survive to reproduce and thrive in that watery environment. Besides looking at like just the anatomy of organisms and their adult forms, we can actually look at homologies in the form of embryos. And so we call this study comparative embryology. So comparative embryology shows that there are homologies not visible in the adult organisms that are only visible in the embryos. Here's an example comparing a chicken embryo and a human embryo. And it turns out that in all vertebrates, embryos have two traits. They always have these two traits at some point during their developmental process. One of those traits is a tail. And so if you look at this human embryo, there's two front arms right there and two back legs right there, and right there. And then they have this tail. And the anus is located right about here, so we call it the post-anal tail. If we look at our chicken, we have the same thing. We have this very long post-anal tail. Now, if you've ever seen a chicken, you know that chickens don't have very long tails. And if you've ever seen a human, I hope you've seen one, you know that humans don't have tails at all. And yet, in our embryo stages, we have these very long tails. And so that's strange, and you might ask why. Another trait that we have in common are these pharyngeal arches, and that's how you pronounce this word, pharyngeal. And pharyngeal throat arches are structures that, if you were a fish, would develop into the structures that become the gills, or the gill structures. Now, chickens don't have gills, and nor do humans. So how did this work? How did this all happen? And of course, if you think about the way that Darwin thought about evolution, vertebrates first, existed in oceans, and eventually, given enough time, they made their way onto land. But the genes of the fish, they still exist in one form or another in our bodies. So let's take our tail, this tail in humans that exists in the embryo stage. Well, you still do have these bones, the tail bones, they just happen to be inside of your body. Turns out that you also still have these pharyngeal arches, but they've been changed and modified over evolutionary time. And now they're structures that are found in your jaw and in your ear and in your inner ear. So these structures have been modified, just like Darwin said, descent with modification. They were no longer used as gill support structures, and instead are used as ear structures. We no longer have a long post-anal tail that has been shortened and exists inside of our bodies now. We could also look at this concept of vestigial structures, and yes, that's how you pronounce this word, vestigial. Vestigial structures are any remnants of features that served an important function in your ancestors, but do not currently have a function. A good example of this is seen in snakes. Snakes arose from ancestors with legs. Their closest relatives are lizards, and lizards have legs, but snakes don't have legs. But if you look at the skeleton of some snakes, you will see It's a vertebral column, just like you would expect in a vertebrate. And you will see a pelvic bone, and you will see bits, just a little bit, or remnants of a limb bone. That's that one bone that's connected to like the shoulder area. So we see those limb bones in snakes. Why in the world would snakes in this location of their body have leg bones and pelvis bones? And the only way that really makes sense is in light of evolution and realizing that they descended from some ancestral lizard and went about their merry way, losing their legs. Now again, Lamarck wasn't right. It's not about use and disuse. It's about whichever feature gives an advantage, that's the feature that survives and allows that organism to reproduce. We could also think about homologies in the form of molecules. So molecules exist like proteins and DNA and RNA, all of these kinds of molecules that have some inheritable aspect to them. They absolutely exist, and we can look at homologies amongst organisms. Just one of the things that you can remember is that all living things have DNA, RNA, and proteins, right? You can think about it that way. You can also think about it in terms of how similar those sequences of amino acids and DNA nucleotides and RNA nucleotides are to other organisms. It turns out that some of the genes that are found in humans are also extremely similar to the genes found in bacteria. In fact, many organisms have retained genes that are kind of like vestigial structures. They've lost their function, but the gene still exists. A great example of this is found in chickens. Chickens have the gene for teeth. And yet chickens do not have teeth. Birds don't have teeth. But their ancestors, the dinosaurs, absolutely had teeth. Chickens have the gene for teeth, but that gene is not turned on. There's no promoter there. So the promoter is turned off, but the gene still exists, which is kind of interesting. If we look at this graph right here, we're specifically looking at hemoglobin. And it turns out that all of these animals have hemoglobin, but if we compare them to each other, we can see how similar these organisms are. So this one's interesting because humans all have hemoglobin, right? So we say of all the variations of hemoglobin that exist in humans, that's 100% human. If we look at rhesus monkey hemoglobin, it turns out that about 95% of the A's, T's, C's, and G's in their DNA, or we could actually look at amino acids because I think that's what this graph is focusing on. 95% of their amino acids are exactly the same. In mice, it's 87% compared to humans. In chickens, it's 69%. Frogs have 54% common amino acids with humans, and this lamprey, which is an extremely weird looking fish, still makes hemoglobin, and the amino acid sequence is 14% the same. So we call those molecular homologies. We all have hemoglobin, it's just that hemoglobin varies between the descendants. The last concept that I want to talk to you about before I move on to a slightly different concept, which is we'll move on to the fossil record soon. But I want to talk to you about the concept of convergent evolution, because so far we've been talking about homologies, and homologies are traits that are, you have these traits in common because a common ancestor had the traits, but it doesn't always work that way. Sometimes there's this concept called convergent evolution, where instead of descent with modification, making things more and more dissimilar, convergent evolution is how things evolve to look similar to each other, because even in distantly related groups, because they've kind of settled on the same solution. to a similar problem. So if you're a flying squirrel and you're trying to get from one tree to the next, it might be really great to have a little extra flap of skin there, allowing you to get a little more air and uplift to hold you in the air a little bit longer. Same thing for sugar gliders in Australia. They have this little extra flap of skin that allows them to jump from tree to tree a little bit better. Again, I don't want you to think that, flying squirrels needed to have this flap of skin to survive. That's not how evolution works. Remember, evolution works because those slight variations that existed, some of them survived to reproduce better. So having that flap of skin that was a little bit bigger allowed them to survive the jump from one tree to another. So that's how this evolved. But the idea of convergent evolution, where things that are not closely related start to appear like they're very similar, they develop traits that are called analogous traits, not homologous. Those traits are called analogous because they don't have the flap of skin here because the common ancestor did. So it's not homologous. We use the word analogous for any feature that's found in distantly related groups, but they live in similar types of environments, or they live in similar ways. And so they've evolved these similar solutions. Convergent evolution does not give any information about the ancestry of those organisms. It just talks about the fact that they have evolved to look somewhat similar. The next big topic that we're going to talk about is the fossil record. And fossil record really does document the pattern of evolution that we see. It provides just a ton of amazing evidence that species have changed through time, and that, in fact, that many species have gone extinct, like Cuvier talked about. Fossils shed light on the origins of new groups and organisms. So here we have the evolution of cetaceans. Now, we already talked a little bit about the relatives of cetaceans above when we were talking about manatees. So another branch off of that group of cetaceans are the whales, dolphins, and porpoises. And whales, dolphins, and porpoises, these cetaceans, they evolved from a common ancestor that includes the even-toed ungulates. Now, even-toed ungulates is a group of organisms, you probably have never heard this term before, that include the hippopotamuses, pigs, deers, and cows. So if you look at most mammals, right, most mammals have two bones right here, and they only have one right here. And this is again on their limb bones. So if you look, however, at the cetaceans and the land-dwelling even-toed ungulates, you can see that they have two bones here, just like most mammals, but they actually have two bones down here. So Pachycetus is an ancestral whale. Sus, or pigs, have two bones, deer have two bones, and if you look at whales today, they also have those two bones in their fins and flippers. So that's, again, more, it puts the lumps kind of together as having a more recent common ancestor, the even-toed ungulates and the whales, whereas most other mammals go and are having the odd toe, like one-toed. Or odd-toed, like five in the case of humans and dogs. So fossil records can show important steps in transitions from one organism to another. So we were just talking about the even-toed ungulates and their transition from the land into the sea. We're talking about their bones, right? The bones in their limbs. So if we put this into this called a phylogeny, I think I've mentioned that before. If we think about that ancestor to all of the other even-toed ungulates, like the deer and the hippopotamuses and the pigs, right? And then if we kind of map on similarities of these fossil organisms like Pachycetus or Rhodocetus. And then if we look at our living cetaceans like this whale here, you can see like this slow change over time from having full limbs to having limbs that are kind of more look like they're for swimming, to having limbs that are practically disappeared in the pelvis, even though we have these ones up in the front of the body, that they're almost absent, although they do still exist. Even in whales, we have the pelvis bone that still exists in the kind of rear end, towards the rear end of the whale. Fossil evidence shows us that living cetaceans and their close relatives, the even-toed ungulates, are more different from each other today than were the early cetaceans and the even-toed ungulates. So that's showing us in the fossil record this kind of slow change that we would expect if evolution did occur. The last major topic that we're going to hit is biogeography. Biogeography is such an interesting topic, and we don't have too much time to go into it. But basically, biogeography is the study of the geographic distribution of species, and it also provides evidence for evolution. So distribution, what does distribution of organisms have to do with evolution? Well, one thing we know is that the continents of Earth used to be a single continent called Pangaea. And those continents that, continent of Pangaea broke apart, and we've got, the drifting of the continents farther and farther away from each other. And so we see this kind of separation of the major continents from Pangaea. But if we look at the distribution of fossils along Africa and South America, we can look at distribution of fossils. Here we see a whole set of very, very similar fossils. At the tip of Africa, the fossils there are very similar to those at the tip of South America. So you might ask why? Why does that exist? And we can say that, well, it makes a lot of sense. Pangaea makes a lot of sense. These continents fit around each other so interestingly. And of course, species, had ranges that spanned the current continents when they were all one continent. I mean, that makes a lot of sense. So biogeography, again, shows us the distribution of organisms. We can look at it the level of fossils, but we can also look at it at modern day species like Darwin did. Remember that Darwin looked at the birds on the Galapagos Islands, and what he saw was that some of those birds, they were only found on those very specific islands, and it seemed like they weren't found anywhere else. And it also seemed like some close relatives might be found on the mainland of South America. So what biogeography shows us is that, we see this over and over again on islands. There are lots of islands have species that are endemic, again, meaning only found there, nowhere else in the world. And that those endemics are related to organisms on the nearest mainland or on the nearest neighboring island. Again, Darwin saw this himself, and we see this over and over and over again as we look at island biogeography. Darwin said that these species arose as a result of these colonists adapting to the new environment, and again, we see that over and over and over again in islands that exist today. So we use this term theory when we talk about Darwin's view of life, right? And I just want to remind you that in science, a theory accounts for many observations and explains and integrates a wide variety of phenomenon. That's important. That's not how we casually use the term theory. Oh, that's just a theory, right? The way biologists, the way scientists use the word theory is that it's a broad explanation that incorporates many, many, many observations and has observational power. It also has predictive power. We can predict things from a scientific theory and see if they're supported by new and continuing new accumulations of new observations and new experiments. So when most people say the term theory, really what they're kind of saying is they're saying, oh, that's a hypothesis. That's a possible explanation. But in biology, a theory tends to have a lot of weight behind it, especially the theory of evolution. The theory of evolution by natural selection is supported by just a huge number of experiments. It's supported by fossil record and biogeography and homology. So very diverse regions or areas of biological study all support this concept of evolution. And this research is ongoing. Many new biologists are coming up and discovering amazing new things, and it's just a continual process of discovery. So that's all I've got for you today on chapter 19. I hope that you enjoyed learning a little bit about Darwin. We're going to continue to learn about him and his ideas and how they're applied these days as we continue on to the next few chapters.